Hello friends, welcome to BMH Learning. In this video, we are going to talk about lipid extraction by Bligh and Dyer method. Lipid extraction by Bligh and Dyer method is one of the standard procedures to isolate total lipid fractions from biological matrices based on a solvent system consisting of chloroform, methanol, water. The basic principle for the lipid extraction is that a biological sample, which can be tissue, blood or plasma, is homogenized in a monophasic chloroform methanol water mixture. After this, a phase separation is induced by addition of chloroform and water. So basically, we use a ternary solvent system composed of both a polar as well as a non-polar solvent. The process is mainly consisted of two steps. In the first step, a solid liquid extraction is carried out from an initial amorphous gel state. This solid liquid extraction is followed by a second liquid-liquid partitioning step. In this liquid-liquid partitioning step, the mixture extracted in first step containing all biomolecules is further separated into two different phases: an organic rich phase that contains the total lipids and a water rich phase which has the other compounds like sugars, proteins, etc. Now this is very important. The crucial point in the second step is the partial miscibility of the chloroform in the water rich phase and that of water in the organic rich phase. So how this property is useful to us? The thing is that the partial immiscibility ensures that all biomolecules, independent of their volume and hydrophilicity or hydrophobicity, are solubilized either in the organic rich or in water rich coexisting phases. Procedure. First of all, let me show you something. This would help you to design your experiment in a proper way. So the table below shows proportions for different volumes of sample. That is how much volumes of solvents you need for particular sample volume. Let's suppose we are doing it for 1 ml sample. So that means we are gonna use these volumes. So for each 1 ml of sample, add 3.75 ml 1:2 chloroform methanol and vortex well. After this, add 1.25 ml chloroform and vortex well. Finally, 1.25 ml distilled water is added and vortexed properly. Then centrifuge at 1000 rpm for 5 minutes at room temperature to give a two-phase system. That is, a system having aqueous top and organic bottom. Now, how you are going to recover the bottom phase? So for this, insert pasture pipette through the upper phase with gentle positive pressure. This means light bubbling. This is done to make sure that upper phase does not get into the pipette tip. So once the pipette tip is at the bottom of the tube, the bottom phase is carefully withdrawn through the pipette. Keep in mind to avoid interphase or upper phase, and you should only try to recover around 90% of bottom phase, not all of it. This was all. Thanks for watching.